Hello, welcome to another episode of BrainCast. This is Marcus. I'm joined today by Professor Leon Lagnado. He's a professor of neuroscience at Sussex University, co-convener of the Sensory Function and Computation module, and a fellow of the Academy of Medical Sciences. Leon, thank you for joining me.
Hi, Marcus.
Hi. So, having a look at your background, you studied mostly physiology. What got you into neuroscience in the first place?
Well, actually, I studied as a medical student at UCL, and I was finding I wasn't terribly enjoying the medical course. The bit I enjoyed most was physiology, and especially neurophysiology, which was and is very strong at UCL. And towards the second year, I was thinking of dropping out of medicine and doing a degree in physics. But then the opportunity to do an intercalated degree came up. So I did a physiology degree, and at UCL I chose mostly the neurophysiology modules that were available. So what drove that, I think, was I liked quantitative science. Physics was my favourite subject at school. And then when I got into medical school, the aspect of what we were teaching that most kind of gelled with quantitative study of biology was the neuroscience, the neurophysiology especially. So, for instance, we learned about the Hodgkin-Huxley model of the action potential, which is one of the exemplars of applying maths to biology. And we learned about that in detail. We learned about how synapses work, and Bernard Katz's work on the basics of how synapses function. And again, that was a beautiful example of understanding the physics that underlies how nerve cells work, and also applying mathematical descriptions of basic processes related to brain function. So I found that very exciting. Yeah, so that's what got me into neuroscience.
Interesting, yeah. And so what sort of stuff now do you work on? What sort of projects have you got going on at the moment?
So I've always been interested in how synapses work, and I've all through my career also been interested in vision. So we work on synapses that transmit visual information. And it turns out that early in the visual system, in the retina, these synapses are quite special. They're quite different to other synapses in the brain. And what I'm really excited about is that we've got to a stage. So when I started working on these sensory synapses, they're called ribbon synapses, way back in the early 90s or mid-90s, the best we could do to try and investigate how they worked was to isolate neurons from a retina and then, say, patch them with an electrode or image them under a camera. And we were able to study how these synapses kind of worked as machines. But we weren't able to study how these machines worked in situ, in the retina of preferably a live animal. But we are now at that stage. So we can look at, we study zebrafish, live zebrafish, and we're now at a stage where we can not only watch the operation of these synapses, but actually get down to a level of description which is getting down to the essence of what synapses do. So synapses transmit information, and it gets back to trying to be quantitative, trying to use maths and measure things. If you want to measure information, the basic framework, the only fundamental framework that's available is something called information theory. And so what I'm really excited about now is that we have the ability to measure the amount of information that visual synapses transmit. First of all, by making very high resolution measurements of how these synapses release their neurotransmitter glutamate, and then by applying information theory to those measurements. And those two things together, if I'd known we'd get to this stage where I started working on these things in the early 90s, I would have been very, very excited. anyway, and I'm still excited now. So yes, that's the most exciting thing. And we're extending that beyond the retina. We're asking about how much information different types of the output neurons of the retina, the ganglion cells, and we're asking about how much information about a visual stimulus they transmit to different parts of the brain. Yeah.
So how do you look at these synapses working in zebrafish?
Okay, yeah. So one of the basic techniques we use is something called multiphoton microscopy. That's a bit of a mouthful. It's a technique which has become incredibly important in contemporary neuroscience because it allows you to look at the activity of many neurons simultaneously in a live animal, a live awake animal. And when I say look, I literally mean look, because we make movies through the microscope of the activity of the neurons. And the way we watch the electrical activity is by genetically modifying the animal and making the neurons express proteins that fluoresce. And there's two basic types of fluorescent protein that we can use. We can use calcium-sensitive fluorescent protein. So it turns out whenever a neuron is excited, the calcium levels go up. So in our movies, the neurons light up when they become excited. The other really important reporter that we use is a reporter for glutamate. So glutamate is the most important excitatory transmitter in the brain. And there's a fluorescent protein that's been engineered to increase its fluorescence when it binds glutamate. And so with that particular protein, we can watch the output from synapses in the live animal. So multiphoton microscopy is very important. We are increasingly trying to relate these very direct measurements of neural activity and synaptic activity to behavior. So we also do behavioral experiments with zebrafish, where we look at responses of the whole animal to visual stimuli. So there are certain reflexes, for instance, the optomotor response is a response where the fish is trying to stabilize itself in the visual environment. That's one example. There's other responses such as prey capture, where the zebrafish have a very kind of stereotypical I don't know if you'd call it a reflex. It probably is a reflex. When they see something that they think is food-like, which is like the size of a paramecium roughly, there's a very stereotypical response where the fish kind of gets a bit under the prey, fixates it a certain distance from its head, and then kind of makes a gulping motion to suck the food in. So that's prey capture. So yeah, so we can relate neural activity, synaptic activity to behavior. And yeah.
So you compare these two elements for in the same experiments, or in the same
yes,
papers?
not yet. That's absolutely right. So so far what we've done is we've looked at how the retinal network or the downstream circuits from the retina respond to certain visual stimuli. And then we've separately made behavioural measurements of fish to the same stimuli, but we've not done it directly together. But I hope soon we're going to be doing that. We're going to be observing the neural activity and synaptic activity, whilst also monitoring the behaviour directly. You can do that with zebrafish, yes.
You can observe the behaviour while monitoring directly.
Yes. Yes.
My understanding of, say, multiphoton microscopy would be that you'd have to have quite precise lining up of the technique. So how would you do that whilst allowing it to exhibit behaviour?
Yes. So that's a very good question. What you do, so we're looking into the brain, yeah? So what you do is you fix the head of the fish.
Oh.
Yeah. But you leave its tail free to move. Oh. And whilst the head is fixed, you can image into the retina and into the brain through the multiphoton microscope. But the fish can still generate a motor response to the stimulus that you apply. And we can assess that motor response by watching its tail. So we have an infrared camera, and we make movies of the tail motion. So they're very stereotypical motor actions that fish do. So you can decode the motion of the tail of the head fixed stationary fish. You can decode that into what would happen if the fish's head was not fixed. Yeah, which direction would it be turning in? How fast would it be swimming, for instance? So that's the trick. Yeah. Fix the head and leave the tail free to move.
And what do you think is going to, or what do you hope is going to be the impact of your research here? If you had to
yes, the impact. I guess, we're in this game for a number of reasons, the science game, research game, for a number of reasons. And what motivates us is both internal and external. So the most important impact for me is actually internal, I'd say. It's, there are certain things I'd like to find out. And if we manage to find them out, I'm quite happy. But of course, we've all got egos and we also want other people to know about it, and we have to publish our work. And that's really, really important. There's no point doing experiments and if you don't tell the world out there about what you've done. And I'd say in terms of impact, to be honest and frank, the audience that I'm most concerned about are fellow researchers, that they will value the work and find it interesting and find it useful, follow it up if possible in various ways and so on. I'm not trying to cure blindness or anything like that. I'm trying to understand the basics of how the sensory processes work, especially visual processes. It would be wonderful if some of the basics that we uncover do inform translational areas of neuroscience, for instance, but that's not my primary goal. Yeah. Yes.
What surprised you most about the work that you've done? Is there anything you've discovered that wasn't expected or made the most sense?
Golly. Something we did quite recently, actually. The question is, how does a synapse represent information? How does it represent a visual stimulus, for instance? And the general thinking is that in any part of the brain, it's the rate of synaptic events that encode, say, the strength of a stimulus. But what we found was that in these sensory synapses in the retina, these synapses can do something very, very clever, which is they seem to be able to release vesicles, not just individually, but release them in multiples, 2, 3, 4 or 5 vesicles, basically simultaneously synchronized. And so what this boils down to is that the information about the strength of a visual stimulus, for instance, is being represented by the synapses, not just in the rate at which they release vesicles, but also in the amplitude of the synaptic events. So it's to draw an analogy. If the dogma being that there's what's called a binary rate code representing the strength of a stimulus, so zeros and ones, that would be a binary code. Mm-hmm. What we found is that these synapses actually, you have more than zeros and ones as the symbols. You have zeros going up to about 11s or 12s. So there are multiple symbols being used to represent visual information. That was surprising to us. And we're exploring that at the moment. You can think of this as an alphabet. How many letters in the alphabet with which synapses transmit information. And it turns out these sensory synapses in the retina have an alphabet of about 12 characters.
Wow. So these the actual photoreceptors that are then outputting these 12 characters.
Mm-hmm.
And the other retinal cells?
Actually, we've done most of this in the cells which are postsynaptic to the photoreceptors. So, So the first step in vision involves the conversion of light into an electrical signal that occurs in the photoreceptors, and those have special sensory, these specialized driven synapses. But actually, we've been mostly looking at the neurons which receive the signal from the photoreceptors, the bipolar cells. We're going to get back to the photoreceptors. I'm not sure it's as simple that how those synapses work is quite how the bipolar cell synapses work. Okay. We're going to look at that.
Could you pinpoint your most interesting or influential paper that you've read or encountered that has affected the way that you work or the things you're interested in?
Yeah, I think I probably could. I think actually there's two. I think they're both papers are from 1953. One is Hodgkin and Huxley's description of the action potential, which was published in 1953, for which they won the Nobel Prize. And then the other paper was a paper by Bernard Katz analysing synaptic transmission at the frog neuromuscular junction and showing that statistically you could describe how synapses work as a particular type of statistical process, a Poisson process. In that paper, they also provided one of the key pieces of evidence that vesicles are the units for transmitting information at a synapse. And Bernard Katz won the Nobel Prize for that as well.
With the amount of things being discovered lately, it seems like we need more Nobel Prizes. And there's going to be a backlog.
Oh, there's loads in neuroscience. If you look at the Nobel Prizes that have been awarded in physiology and medicine, a surprising number actually are in neuroscience.
The brain is the best, according to the brain. Do you have a favourite fact or part of the brain?
Favourite fact about the brain? Oh, golly.
Something you would tell non-neuroscientists and they'd be surprised, to impress them.
Well, one really interesting fact, getting back to the visual system, which I think tends to surprise not just the general public, but students as well when I tell them this, is how many photons of light can a human detect? And the number is actually surprisingly low. It turns out that a human can detect the arrival of just a handful of photons at the retina will reach consciousness. Will be perceived. Yeah,
that's there's a lot of photons that could fit in my hand, I imagine. But you
fingers, count the fingers, Marcus. Yeah, 5 or 6, something like that. Photons. Wow. And actually that's so few photons that no more than one photon hits any individual photoreceptors. So that means that at the individual photoreceptor in your retina, a single rod can reliably detect a single photon. Yeah, that's the ultimate. You can't get smaller than a photon.
Yeah,
and that's the ultimate. And so the sensitivity of our retina. This is only after you've been sitting in a dark room for an hour. You have to adapt to the darkness, but then your visual system gets to the ultimate sensitivity. Wow. Individual photons. And actually, I did my postdoctoral work with a guy called Dennis Baylor, who was the first to demonstrate directly that rod photoreceptors could indeed reliably signal the absorption of a single photon. Yes.
Can we? I don't know if it's your field of knowledge here, but could we do experiments, or do we know about other things the eye can do at that point? If we can see individual photons, then could we see things potentially smaller than we could, or a better resolution than normal, if you don't have all the other noise going on there?
The resolution in space of what we see it's basically determined by how closely packed together the individual photoreceptors are. So to see detail, we use a part of our retina called the fovea. We have to focus our fovea. When reading a book, we're focusing our fovea to make out the letters. And what determines your spatial resolution is how small the individual photoreceptors are and how closely packed they are. And actually in the fovea, the photoreceptors tend to be thinner and they're very, very, very tightly packed. I'm told that in some animals, like birds, they're even more highly packed, so that they have eagles, for instance, have higher spatial resolution regions than us. But that's the limiting factor for spatial resolution. And actually, to use that spatial resolution, you need relatively high light levels. You can't do that at low light levels where there are very few photons around. And one reason is because in our fovea, the photoreceptors that we're using aren't the most sensitive ones. So I guess there are rod photoreceptors and there are cone photoreceptors. Yes. The rods are the most sensitive, and we use them at night, at dusk, and so on. Mm-hmm. In this room right now, we're mostly using our cones, and they're less sensitive. Yeah.
Okay. Has anything you've discovered or your own research, has that changed or the way you go about life in any certain ways?
I'm not sure that there's any particular discovery that we've made, advance in understanding that has affected my life. But I would say has almost certainly, I'm not sure of all the ways, but almost certainly will have affected my life is more the way we do research. So when you do research, there are times when you have to learn to be patient. There are times where you have to cope with disappointment. There are times when you have to have the discipline to be methodical. And I'm sure that's spilt over from my working life into my general life. In the lab, I do a lot of dealing with the equipment, repairing it, making sure it works and stuff. And that kind of tinkering thing that I do in the lab, I also do outside the lab. So I think it's more about an approach to problem solving, but also an approach to dealing with ups and downs, because doing research, there are ups and downs. It's there's the excitement of finding new stuff, and that's wonderful. But then, you get papers rejected when you send them to journals, and, you have to deal with that as well. So that experience must have affected how I deal with things outside the lab. I'm not sure of all the ways, but yeah.
And I guess a final question, a little silly one. If you could upgrade a part of your brain,
yes,
what would it be?
So in the mathematical part of my brain, I think. So I didn't have any formal maths teaching beyond A-level maths. And after then, it's all been self-taught with textbooks and picking up the math skills that I felt I needed for particular problems. So I'd love to have slotted into my brain a more comprehensive, well-founded, general understanding of a bunch of mathematical concepts, some of which I only half understand and am not in command of at the moment. So yeah, that would be it.
Well, thank you very much for all your time.
Oh, yeah. Pleasure, Marcus. Thanks for
very
having me.
Cheers.